Hello, this is Katie Butts-Dehm, your biology instructor. Today we're going to do the lab on osmosis and diffusion. So the first thing you're going to want to have is the lab handout, osmosis and diffusion. And the second thing that you'll want to have is the answer sheet for osmosis and diffusion, or diffusion and osmosis lab. And you're going to turn the answer sheet in for a grade. The lab sheet has the instructions. Let's see. So here's the lab sheet, the osmosis and diffusion lab preparation, or diffusion and osmosis lab preparation. So here that is, and you need to make sure that you have everything here. So 2 or 3 chicken eggs with the outer membranes intact, but the shells dissolved. And there will be instructions on how to make these later, how to prepare the eggs. You actually should do the eggs about 3 days beforehand. It'll take about 3 days to remove the shell using vinegar. Then you need 6 cups, clear cups from the lab kit. The ones we used in the lab kit work just fine. And there I have these clear cups. You can use cups at home too, like glasses if you have any. You need the 300 milliliters of 15% NaCl. You actually need, this should be 500 milliliters actually. So 500 milliliters of 0.9% NaCl. Use table salt for the NaCl because that's what it is. 300 milliliters of distilled water. Now, tap or bottled water should work just fine as long as you use the same water to make your other solutions. And distilled is always the best, which I would use if I were making a lot of them for, a bunch of lab students or for a whole class, but this will work just fine. So tape and Sharpie or something to label the cup with, any permanent marker, salt, an electronic balance, and let's see, electronic balance. That's a little one from your lab kit. Weigh boats, and either the weigh boats from the lab kit, but something like Dixie cups will work, or a piece of wax paper will work for the salt. And the lab handout and answer sheet, a clock, a calculator, and you can use your cell phone for both of those. A medium-sized potato, a knife to cut the potato, a cutting board or plate, paper towels, celery or Napa cabbage, and a cup or a glass, and another cup or a glass, food coloring, water, a non-serrated knife or scalpel. So not one of the knives with the jagged edges, but of even one. And, like this one here. Let's see, and a cutting board or plate, saran wrap, and vinegar. So we're going to go ahead and make the first solution, and I'm going to work off my own sheet, so that way you can see what's going on. Okay, so we're going to make 300 milliliters of 15% NaCl solution. So, I have some, but I'm going to make some more. And what I need is an empty container. And these plastic bottles now are so flimsy these days. So there it is, an empty container. Hopefully it will become less flimsy as I put stuff in it. And then we need, let's see, 45 grams of NaCl. And 300 milliliters of water. Now the thing is, your entire solution has to end up with 300 milliliters. So what you're going to do is you're going to add 250 to the salt, and then you're going to check the solution, and you're going to top it off at 300 milliliters. So you're going to want to make sure that you label your solution And I have some masking tape that I put on here, so I guess I'm going to actually use this one. And that's going to be 15% NaCl. So make sure you label it. If you work with other people, you always put your initials and always put the date. You're included, because you never know what you're going to find in some lab. You need to know how old it is. And so what we're going to do is 45 grams of NaCl. So I'm going to zero this out. And I know for me, I can read the zero. Oh, you can see the zero. In the past, you haven't been able to see the zero on this. And here's my salt. So I'll just add. We have 16.89 grams. 22. 33. 39. 40. 43. 44.98. Oh, it's 45. 45.04. So we're going to call that close enough. So we're going to go ahead and put it in here. And now we're going to need water. And remember, it's going to be 300 milliliters total, but we're going to start with 250. And I'm just using the same bottled water that I use for my distilled water. So we have, it's going to be 50. Now there's still some salt in the bottom of that funnel, but that's okay. The water will get that out. Now, if I were doing this at work, what I would do was use a bigger funnel. Okay, that's 50. That's 100. That's 150. That's 200. And here's 250. Now remember, we want to end up with a total volume of 300 milliliters. We have 250. And we don't know how much volume the salt took up. So go ahead and make sure this is all mixed up. Set this aside. And then I'm going to go ahead and get my empty container. And we'll check the volume on this. And make sure the total volume is 300. So here's 50. $100. Here's 150. Little too much. Perfect. 200. Here's 250. And we needed a total of 300. So let's see what left in the bottle. That was the volume of the salt. Okay, so we used about almost 20 milliliters. And now we'll just take this with our plain water and fill it up to the 50. And that is going to make it be 300 even. And then I'm going to put it back in here, the labeled bottle. It will be a little bit more stable once it's filled with water. I would just leave it in the squishy bottle that I have, but it's so squished up that I would rather just put it in here. And I could pour it directly back, but there was a little bit of salt in the bottom of that. It's gone now. So here we go. So this is our 300 milliliters of 15% NaCl, okay? It's all made, all ready to go. Now, one thing I'd like to tell you is you're also going to have distilled water, which for the distilled water, again, tap water, if you use tap for all of them, or I'm just using this bottled water for all of mine. So, you know, it works best with distilled, but it'll still work just fine. The other thing that I wanted to show you was the next solution we're going to make, because I'm not going to make them all on here. But anyway, let's see. Okay, here it is right here. The next solution, that solution was 300 milliliters. So the next solution that you make is going to be 500 milliliters. So make sure your final solution is 500 milliliters. So this first solution that we made is going to be a hypertonic solution. That means the hypertonic solution has more solute in it than a cell. And we'll look at that a little bit later. But it has more salt. Salt is the solute in this solution, and water is the solvent, okay? And this one is going to be an isotonic solution. So the 0.9% NaCl, you should have about the same percentage of solutes in the cell that you're going to compare it with. And then for the distilled water, that's our hypotonic solution. There shouldn't be any solutes in there. So that is why that one's a hypotonic solution. So the next thing you're going to do is you're going to prepare the eggs without the shells. And this is really important that you wash your hands after you handle the raw eggs. When you're handling the eggs and vinegar, you should wear gloves and safety glasses or glasses like this with some shields, side shields. And you don't want to get salmonella though from handling the raw eggs. You're going to put each egg in a cup or glass, and I just use the plastic ones from your lab kit. Again, something like this, okay? And just make sure, just a plastic cup. You put the egg in it, and then you put vinegar in the cup, and you'll cover the top with saran wrap. Put the cup away from your pets and your children so none of them can get it. You're going to let it soak for about a day. You'll pour out the old vinegar and you'll add the new vinegar. And basically you can see immediately when you put the vinegar at the beginning, you see all these bubbles form. And remember the chemical reactions we saw where the vinegar reacted with something and then CO2 was released as a gas. So you have the gas released and the calcium carbonate eggshell is going to be broken down. So you switch out the vinegar after about a day, and then the shell will end up being removed. It should take 2 to 3 days of the egg soaking in vinegar. So an hour before we start the experiment, I'd like you to place all 3 of the eggs in 0.9% NaCl and then let them sit for an hour. So the good thing about that is they'll all be starting at the same place in an isotonic solution. So the eggs, I did my eggs in the earlier video you might have seen, but they weren't ready for then, so they are ready tonight. So make sure you read all the background information on the sheet, and then we're going to start with this lab. So this lab is just the first experiment, or the first experiment. It's just a little experiment on looking at diffusion, and it's pretty simple. So diffusion is the movement of molecules from a high concentration to a low concentration. So I will switch this over here. There we go. And what we're going to need is any clear glass like this, and then you're going to put some water in it. I was worried we brought too much water here, but I'm going to put the water here. And then all we're going to do is pour some food coloring in it. And see what happens. So molecules are always moving. Molecules always have energy, and they're always moving. Maybe we'll use purple. And that's why they move. Now, when you do this, my water's not settled because I'm going to have to move it out of the way and everything. So I'm going to end up not settling it anyway. So when you do this, set your water up to where you're not going to be touching it. And then you'll just put a little food coloring in it. And right now you can see the food coloring is very concentrated in one area. So the food coloring molecules, without you stirring it or anything, are going to gradually move from the high food coloring concentration throughout the water to lower food coloring concentration until the concentration is even. Also, the water molecules are going to move from high water concentration to lower water concentration until that solution is going to be even. So this I'd like to share with you something, and I believe it's this one. I'll just share with you the screen since it's not going to want me to do that. So this right here, if you have this higher concentration of dye in the water or salt or whatever solute it is, and the water's a solvent, then it's going to spread out to where the molecules are eventually going to be like even, whether you stir it or not. And see, here's what happens. And that is diffusion. So if I did not touch that cup over there, then it would be, you know, in an hour or less, it would be back to, it would be all even. Right now, it's not. And actually, this food coloring looked heavier than a lot of food colorings I use. So I thought that was sort of interesting. Let's see. So this is what we're doing right now in this experiment. And you just sit and wait, and there you go. Now, another thing that I wanted to point out is osmosis. So this is osmosis. This right here is a cell membrane. It is made up of a phospholipid bilayer. These are the hydrophilic heads on either side and the hydrophobic tails in the middle. So osmosis is a type of diffusion, but it's diffusion of water through a selectively permeable membrane. Now, selectively permeable or semi-permeable means it allows some things to cross through it, but not others. So the water is going to move through a selectively permeable membrane from higher water concentration to lower water concentration. And you can see the little solute, by the way, that looks like glucose, something like that. But this side here has much more water and less solute, okay? And then so the water is going to move from high water concentration through the membrane to low water concentration until they are evenly distributed on either side of the membrane. So for osmosis, it's a type of diffusion, but you need to have two things happen for it to be osmosis. It's diffusion of water and it's through a membrane. So that's what makes it osmosis. Okay, so we're going to start now. We still have this one. And what I should have done is looked at the time at the very beginning. But right now it's 7:35. And we'll just look at it every so often and write down our observations. And have a result section on the answer sheet down here. And I can just write down the time and what I see. And the date, if you know, it should be all the same date. And my food colorings at the bottom. And then I'm going to observe it every so often. And there it is again, still at the bottom. Okay, so we're going to start the potato experiment and start to set up. So a lot of the experiments from this week, they have set up and then, so you'll set them up and then you let them wait. So we have 3 cups, let's see, obtain 3 cups. And these are the ones from the lab kit right here that I washed and rinsed out. Label one of them distilled water, or DH2O. The other one, 0.9% NaCl. And the other one, 15% NaCl. Okay. Now what you're going to do is you're going to add 100 milliliters of each liquid into the cup named for that liquid. Okay. So I'm going to start with the distilled water because that's not going to change the concentration of anything. Everything's made with the liquid and with distilled water. So here we go, distilled water, 100 milliliters. And honestly, for this experiment, it doesn't matter that much how exact you are. You just want to make sure they all have about the same amount. So that looks pretty good. That's distilled water. And then the 0.9%. Make sure you put that in the right cup with the right label. There, it's the same amount as that one. And then the 15%. goes in the other cup. And make sure the 15% is well stirred, so that it's dissolved. And double check. Make sure they all look like they have the same amount. Yes, they do. So here we go. There's our solutions in our cup. And then we need 3 slices of potato. And we want the slices of potato to be very similar to each other. Now, I don't want any of the peel on it, and you can peel the potato if you want, but I don't want to be touching it and, you know, everything like that. So this is just faster for my purposes right now. Okay, so 3 pieces that are of approximately equal size. Well, and they don't have any peel on them. You'd think I'd never cut a potato before. For some reason, it's bigger in the front part. Okay, yeah. 1, 2, 3. I'm going to just cut another one 4. And just see which ones of these are going to weigh a similar amount. Okay. This one, great. So you're going to want to use your weigh boat and weigh them and make sure that you record it. So you zero out your weigh boat, and this potato slice is 14.54 grams. And for that one, plan on putting that potato in the distilled water. So I'm also going to write it down, but 14.5, I'm going to 53 grams. Okay. And that one's going to go with the distilled water. All of these are sulfur in this. And make sure that's zeroed out. The next one is 17.76 grams. How can that be so much bigger? Let's try this one. 17.61. I'll look back at this one. 14.56. Well, that's pretty small compared to the other. Oh, 18.55. Try this one. 18.06. You know what? This is 17.65. 7.63 now. 17.63. 17.63 grams. Is a distilled water one. And then the next one we have 17.72 grams. Much closer. And then the next one is going to be 17.56 grams. And that one's going to go here. So we're going to go ahead and make sure that we write them all down. And let's see, 17.63, 17.72, and 17.56 gram, gram, gram. And the third one is in the 15%. Great. And then we'll look at the time. It is 7:48. Oh, it's 7:49 right now. 7:49. Okay. And we'll just take them all and put them all in. 1, 2, 3. So there we go. There's our experiment ready to be done. And 45 minutes. What time will it be in 45 minutes from now? So that we need to figure out, because that's when we need to take these out. You know, I almost feel like I ought to set a timer for 45 minutes, but that's actually not a bad idea. So I'll set a timer. Timer 45. So we're starting with that. And also, let's say it's 7:50. So that would be 8:35 minus 1 is 8:34. So it should be about 8:34 is what I'm thinking. Now, one thing I did to keep the experiment controlled, you might have noticed, is what I do, I put all the potato slices in there at the same time, okay? And one other thing too. So we want to look at this real quick. So here is the dye, and look, it's still at the bottom. That's the heaviest dye or food coloring I've ever seen. This is a different food coloring, but it still will spread out. Okay, and then here's our 3 potato slices. And one thing you might notice that some of them are floating and some of them have sunk. This one, the only reason it's floating, should not be floating. Oh, there, it's because it was so big that it was stuck being held up by the cup, okay? The only one that should be floating is the one in the 15% NaCl, and the reason it's floating is because the salt makes the solution more dense than the potato, so the potato floats. Now, what should happen is there's a higher concentration of water in the one with plain water than inside the potato. So water should move into the potato from high water concentration to lower water concentration. So this one in the cup, it has 0.9% NaCl, and it should be about the same in the potato. So some water should move in and some should move out, but it should be about the same amount. Now, this is 15% NaCl in the solution, right? So that means it's 85% water in the solution. So the inside of the potato cells should have about 0.9% solute. So that's close to 1. So they should have about 99% water. This means water should leave the inside of the cells and go out into the solution. So this one, the potato should shrink. It should weigh less, it should shrink. And this one here, the potato should weigh more. And it should be like a little, you know, swollen up and such. So we're going to set these aside, and then we'll move on to the next experiment. Okay, because we have to wait 45 minutes. So the next thing we're going to look at is the egg experiment. So. Okay, so I just have a plate and some paper towels, and these are the eggs that I have. They're right here sitting in, actually they've been sitting in 0.9% water for, excuse me, 0.9% NaCl, about an hour. So I'm just going to go ahead and get rid of some of the water so we can look at them. And they should all be starting, you know, about the same. Now, you know, I've had some people tell me their eggs broke, and I had one break before, but it's weird because when you feel them, they feel very rubbery and they don't seem that delicate, but I've had one break before. And by the way, I forgot what I was going to say, but there you go. There's the eggs. And actually, in hindsight, I wish I would have done this after I got something ready. So I'm going to go ahead and put this back in. But they all should be about the same at this point anyway. Of course, there's individual differences in eggs. So what we're going to do is get 3 cups like before. And where's my 3 cups here? 3 cups. And then we're going to label them. And we're going to have 1 in distilled water and 1 in the 0.9%. So we got DH2O. And I don't know if you can see that, but I can. And then 0.9% NaCl, which was 4.5 grams of NaCl in 500 milliliters of solution. And then we have 15% NaCl. Okay, so we have our cups ready. The next thing we want to do is, let's see, gently dry the cups, gently dry the surface of the eggs, and record the observations. Weigh a chicken egg, record the weight, put it in the cup, record the time. Weigh a chicken egg, record the weight, put it in the cup. Okay, so I guess I'm going to just have to dry them very briefly. And one thing, it doesn't say how much liquid to put in the lab. So we're going to want to put in approximately the same amount and enough to cover the egg. So. There's one egg. There's one egg. And then here is another egg. Our wastewater, and we're going to dry them, but I don't want them sitting around soaking up all of the, or have the paper towel soaking up everything out of it. So I'll just roll them over and put it on this wax paper plate. Okay, and now we're going to take the one that goes in the distilled water. And you know what I think I might do? I might use the cup since it's empty for a weigh boat. Okay, so I'll turn this on. And zero it out. And this is going to be the one with the distilled water. And you want to put it in really gently. And this is 87.93. Okay, so I'm going to write that down somewhere. 87.93 grams. And of course, it should be on our lab too. On the lab sheet, 87.93. Now that I've been handling eggs and touching this pen and everything else, everything's going to have to be washed and disinfected. Okay. So this is the 15% NaCl. I'm going to do the 0.9% next. So we'll zero out this cup. It should be about the same, but you still want to zero it out to make sure. And then we'll put this egg in here. And this one is 88.01 grams. And that's in the 0.9% NaCl. So 88. Now it was 0. Yeah, going back and forth, 0 grams. When I touch the table, it means 87.99 and then back to 88.00. Okay. And then now the next egg, make sure our cup is zeroed out. And here's the egg. Put it at the bottom without breaking it. I feel a little bit safer. This is 86.9695. 86.95 grams. 86.95 grams. 86.5. And the other one was 88.00. Okay. So now we've got all of those ready to go. And then we're going to add the liquid. And this is going to be 45 minutes. It's 8:05. So we'll add the liquids. This is the 15%. 15% NaCl. Oh, look, and it was enough to cover the egg. That's great. And I'm not going to need any more. So they'll all be filled to that amount. There it is. Notice the egg floating. Now we have 0.9% NaCl. Okay. So there, that one's has the water. And now we're going to have this plain water. Now I want to make sure I look at these, see that they have all about the same amount. Well, maybe I got a lot on this one. Well, maybe a little bit on that, more than this, and a little bit more on that. So there we are. So we're going to let these sit for 45 minutes, and that would have been 8:50 plus it's 8:07 now, so 8:52 should be the time that we're going to remove it. Or remove them all. 45 minutes, 45 minutes, 45 minutes. And you know what? You can do this longer than 45 minutes as long as you do them all at the same time. The reason I usually do 45 minutes is I know that it works at 45 minutes, and usually I have 2-hour labs. So with 2-hour labs, you have just enough time to set things up as fast as you can and then let them run for 45 minutes. And the difference, you might be wondering, why are we doing the same thing for the egg as we're doing for the potato? But there is a difference. And the eggs are animal cells, okay, with no cell wall. And the potatoes are plant cells. They have a cell wall, a large vacuole, and so they're going to be different. The results are going to look a little different. And it's really neat to see the egg like that, okay? And just make sure when you look at that, then you are you clean everything off when you're done with some type of disinfectant. Okay, so we have one more activity to set up, and that's going to be the celery or the Napa cabbage. So I'm going to get everything ready for that. Okay, so what you really need is 1 cup, and I have 3 because I just want to do this with 3 cups. But so all you're going to do is put some water in your cups, and then you're going to put food coloring. And so it doesn't work very good with green because it doesn't show up good, but this is a teal. Usually it looks good with blue and with red, but I have a different type of food coloring. Oh, look at that purple. Look at that. So you can see it's really a heavy, heavy purple. And then we've got, I'm going to try this. And you know, you just can't have too much food coloring. So what we're going to do is we have these now and they're out if I need more. And we need to get some slices of celery. And you want to make sure that you're not using a serrated knife for this. You want to get the even knife and just cut the celery. This is the worst knife ever. I hope it's going to work with this bad knife. So I'm going to pause and go get a good knife real quick. Recording. Okay, so I'm not sure how much I showed you here, but I got celery and I put it into these containers, and we had a one in a purple dye that I cut, and then one in a teal dye. And by the way, red and blue work really good. Usually green doesn't. And then we have one in a pink dye. Okay, so I cut them right here. Okay, right over here. And I used a non-serrated knife, and then I put them in there. So we can wait 45 minutes, or they're actually better if you wait overnight. So I did some a few days ago, and I just cut the end of one that was magically here in the pink food coloring. And you can see these little dots here. Okay, those are vascular bundles. Okay, and let's see if we look on this camera, that one seems to be going a little bit blurry. So if you look on this camera, you can see the vascular bundles pretty well. So, and it's xylem is the name of that tissue. So in us, we have veins and arteries and capillaries that are our vascular tissue, and it's bringing our blood. Vascular tissue is transporting that. So plants have xylem, and the xylem, little bitty tubes like blood vessels, but it transports water from the roots of the plant up through the rest of the plant. So this is xylem that you're seeing. Now phloem is the other vascular tissue, and that transports carbohydrates and sugars that the plant makes. It transports that from the leaves down through the stem to the rest of the plant. Okay, so that's phloem, like phloem for food. That's carbohydrates or food that it makes. And then xylem transports water. I believe I have a video here somewhere that shows a little bit of that. So here would be a vascular bundle, and this right here is the xylem transporting water, and that's the phloem. And then there's other tissues in here. And let's see. Oh, here you can see these little tubes. So those are vascular bundles, and it has both the xylem and the phloem. And the xylem's bringing water up. The phloem brings the sucrose, that's sugar, or other carbohydrates down. And here's, how we see it. That's the celery type. Okay, xylem. Oh, this one looks like There's the water moving up the xylem. More water moving up the xylem. And there's food or sugars moving down the phloem, water moving up the xylem. Okay. I need to make that work all together. But here's what you'll see afterwards. Okay, and when you get those carnations that have blue around the edges of them, then that's because of they've done this with it. And the dye has moved up the xylem. This one right here, no, that's not one I did last time. There's the xylem, the vascular bundles. There's also going to be phloem in there, but they run all through the celery, and those are the strings that you get in your teeth. Oh, I think that, I don't know, these look like they're mine because I use Napa cabbage, but they might not be mine. And there we go. So stop share. And now I'll switch to the other camera and we'll look at this celery a little bit more. So you can actually cut down them and see if they run like the length. And that to me is kind of neat. Some people can pull the tubes out. Oh, there's one that's coming out. So look at that. Isn't that neat? And here you can see how the vascular bundle's there. And I wonder, oh, it still smells like celery. Look at that. Isn't that neat? You can pull one out. So there is the dyed vascular bundle. The rest of the celery, notice, is not dyed, but also you can see that it went all the way to the end as well. And that was with the pink, but the pink did really nicely. And then I also did one with purple. So that just looks dyed purple, the whole end, but we'll cut that and see what we have. So you can see the vascular bundles there. There's some way I used to could get this camera to focus. It's only been acting up for like a week or so. And you can see this back here is focused. But that's the same thing. So you can see this really well here. And here it is over here. So you can see the purple really well, the vascular bundles. And if I cut along the top, uh-oh, well, I didn't do that great a job. That turned out good. There. Oh, yeah. And then let's see. See, you can see the vascular bundles. I think I see it a lot better than you do because for some reason it's in the green and the green looks a little dark on the camera. But I can see them really, really well. And actually I can see them a lot better than I could see the red, even though I can see the red really good. Oh, now I have a whole thing of the vascular bundles in here. If I wanted to, I could probably pull them out and see them a little bit better. And then I also did a one of them in the field. So I bet it's probably not going to work as good, but we'll see. It's got to work better than the green. I just thought these food colorings. These aren't the usual food colorings I use. Okay, so you can still see it actually, and they look pretty good. So definitely better than the using green would be. So green or yellow, stay away from those colors, but blue. You know, if people didn't mind, I mean, you got a safe food coloring or something like this might be a neat way to serve the celery, you know, especially if you had like a pink or something at a baby shower and maybe some pink and blue little vascular bundles in it. Wouldn't that be cute and special? Okay, so there's that. Okay, so it's time to take our potatoes out. And one thing that's really important is you do not want to squash potatoes, okay? You don't want to squeeze all the liquid out of them because that would be silly. So we're going to take a distilled water one. You do have to dry them now, okay? Oh, now this is a shame. The one thing I forgot to do is to feel them in the beginning for the texture. But I can take any of my potato slices and feel them. So here's the one in distilled water. And so it is now 18.23 grams. Now before it was 7.63, but now it's 18.23 grams. So it gained quite a bit. So not quite a gram, but it did definitely gain. And that's what it was supposed to do. It was in the plain water. Water was supposed to move from high to low concentration. And when I feel this, it's very stiff. You see, very stiff, very crunchy feeling. Really, I could take off my gloves and feel it a little bit better. So now we'll do the 0.9% NaCl. And this one, okay, well, it's just a little bit more give than the other, but I want to dry it first. And don't squeeze all the water out of it, just get it off the surface. And then put it on there. That's 17.56 grams. You know what? That was 17.72 grams before. That is really close. Really, really close. Okay, so that's good. That one should have lost some, gained some, but basically it should even out. So it's a little bit different. It did lose a tiny bit, but that could have been just because maybe I dried it too much. Okay, and that one feels just a little bit more give. I don't know if you can tell, but a little bit more give than the other. Otherwise, it feels about the same. So we'll take the 15%, and you'll notice before it was floating. Now it's not floating. Now it has sunk. So our scale is still reading 0. And again, don't squeeze all the water out of it. Just make sure you dry the surfaces off. And I'm trying not to move it, but I'll tell you, this one is so flexible, it's hard to not move it. That is 14.79 grams. Before it was 17.56 grams. Now it is 14.79 grams. Okay. For that, oh wait, leveled off at 14.81. So we'll go with that. 14.81 grams. So that has really lost a lot. And check this out. Look at this. Isn't that flexible? Like I was saying, it was hard to not move it. So there's our 3 potatoes. And let's see, the eggs aren't ready yet, though. They won't be for a while. But I just wanted to look real quick at the chart. Let's see. Share screen, share it here, and oh, not that though. And not that, but that is what happened was osmosis. The water moved into or out of the potato. Look at the answer sheet. So you have the pre-lab, which you need to make sure you look at the videos and these links to answer the pre-lab questions. Here's the results. Oh, with the dye, we should have been checking it every maybe 10 minutes. And, but right here are initial masses of the potatoes. We had, in the distilled water, it was 17.63. And then the initial potato texture. And I know that they're crisp. And I was cutting it, and rigid too. And then we've got the time in solution. They were in 47 minutes because I went a little bit above. The final mass of the potato, and the final mass of that potato was 18.23. And I'm just going to get these masses in real quick. Okay, this one initially in the 0.9% was 17.72. And then it was also, they were all crisp and rigid at the beginning. And then this one here was 17.56 grams. And they're all grams. I wish I had fit grams at the top over there. Then I wouldn't have to write it on each one. So I had to put it up in here. But time in solution, 47 minutes, 47 minutes. Like I have minutes up there, see? And then final mass of the potato, 18.23 grams. And then we have 17.56 grams. And then we have 14.81 grams. I think it's coincidence this one ended up at what that one started. But our final potato texture, we had, this one was crisp and rigid. This one was crisp and a tiny bit, it's not rigid, but it's not super flexible. This one was flexible and it is slimy, this one. Flexible and slimy, it's very flexible. The net movement of water, normally I would have one where you add, put the number, and then one where you put plus or minus, but we're just going to do them both. So we're going to add them up here with a calculator. So that one, 18.23 minus 17.63 equals 0.6. So, and it gained the weight, so it's going to be plus 0.6 grams. So this next one, we had 17.72 minus 17.56. So that actually lost 0.16. So it's a way smaller number than the top one there. And this other one definitely lost. And look at this, it's going to be 17.56 minus 14.81. That, look at the difference, 2.75 grams. That lost a huge amount. A huge amount of water came out of there. So, and the tonicity, the first one was hypotonic. When you put cells in a hypotonic solution, hypotonic, then that means it has a lower solute concentration, but a higher water concentration. So water is going to move into the solution. Now this one should have been isotonic. And you know what? It did slightly lose a tiny bit, but not anywhere like that third one. The third one was hypertonic, which meant that it had a higher solute concentration in the solution than in the cell. So this one here definitely lost a lot of weight. So then we have our eggs to look at. And our eggs are going to be done in 5 minutes. So I'm just going to look our initial mass of the egg, 87.93. And then this one, and that's grams. This one is 88.00 grams. And this one right here is 86.95 grams. And they all were rubbery. I don't know if this makes sense to you, but they felt full. Okay, so we're going to go with that. And they should all be 45 minutes in the solution, but we'll see. Okay, so time in solution, 45 minutes. I don't have to write that because minutes is at the top. And then final mass of the eggs. So we're going to go ahead and do that. So, you know what? I want to put these on the right way and I probably should have got a spoon to take it out instead of a fork. But here we go. So we're going to take the egg that's in the distilled water. And again, we don't want to squeeze the water out of these because then it'll mess with our mess with everything. So not a fork. But here it is, the one in the distilled water. And before it was 87.93. And now it's 89.43. Okay. So that one actually gained after, let's see, it gained some 89.43. And it's 8:52. So I need to go ahead and get these all out. Okay. And then the next one we have is 0.9%. Really hard to get out of there. Have an empty container and make sure not to get your eggs mixed up. Okay. These are really fun. So I better weigh it first. This one is 88.47 grams. 88.47 grams. And you know what? It was 88.00 grams initially. So it did gain a little, but not very much. This one right here is still floating. So you know what? I may actually put these back in if I remember. Oh, this one is different. The other two just felt like they did before, but this one actually feels softer, and you can see the egg yolk a lot better in there. And you can see it moving around. The other ones you really can't very well. Maybe it's not moving around. It might be so full of water. Let's see this one. 83.68. 83.68 grams. So it lost actually. It lost, yeah, it did. It lost, it looks like close to 3 grams. So I'm just going to put this back in the 15. and it's back in the 0.09%, but oh no, I'll have to use the potato 0.09. It looks like I don't have any left, so that means it's not going to be valid if I wait another hour and look at it. And then look at this one, but the other two will be still back in the water. So the other two will still be able to look at, and that one shouldn't change. And now we will turn this off and look at those numbers. Get my gloves off. And let's see. So we'll share the screen again. And here they are, the final mass we have. This one was 89.43. And that one was the one in water. And the next one was 88.47. And then the last one was 83.68. I think this will be fun to look at it later. And the way the other two still felt rubbery and full, both of them still did. Okay. But that other one felt, it still felt rubbery, but it felt kind of flimsy. It looked smaller, and I could easily see the yolk. Okay, so the movement of the water, we're going to look at that real quick. And we're going to have 87.93, well, 89.43, 89.43 minus 87.93 equals 1.5. So that's pretty good. This one gained 1.5 grams. So that's pretty good. The next one was 88.47 minus 88. Well, that should be pretty easy. It won, let's see, it gained 0.47 grams. So about half a gram. So it gained a little bit, but a third of the amount of that, you know, under a third of the amount of the other one. And then this one here, we had 86.95 minus 83.68 equals 3.27. I'm going to do that one more time because I forgot to zero after the other one. 86.95 minus 83.68. 3.27. So that one is minus 3.27 grams. Okay, and so definitely this one lost a lot. The 15%. The other one in the distilled water gained a lot, and the 0.9% NaCl did gain a little bit, but not near as much as the other one. So this should be, the distilled water should be hypotonic, as the distilled water had less solutes. The 0.9% NaCl should be isotonic. And the 15%, and you know, this might actually could be affected by how old the potato is too. Isotonic. And then this should be hypertonic. The 15% salt is way more solute in that 15% salt than there was in the potato. So this is the end of our lab tonight, since we already looked at the celery results. If you have any questions, feel free to email me. Any questions about how to do the lab or about the results. If your results don't work out just right, exactly like you expected, then that is fine. You just let me know. Let me know what results you got and then why you think that it happened that way, and that will be fine. So I hope you understand diffusion and osmosis a little bit more and have a good night.